Hello, this poster presents our work on an integrated consensus-based approach to characterize alternative splicing in peripheral arterial disease, or PAD. Alternative splicing is a powerful mechanism to increase proteomic diversity in cells and tissues to adapt to various conditions. In humans, about 95% of multi-exonic genes are alternatively spliced. There are numerous modes of alternative splicing observed, of which the most common in humans is exon skipping, which is about 50% of all alternative splicing events, as shown in Figure 1. Alternative splicing is believed to play a major role in development, tissue-specific processes, adaptation to the environment, disease, and aging. And in this context, we focus on PAD, an aging-associated circulatory condition in which narrowed arteries reduce blood flow to the limbs. It is a major cause of muscle damage and mobility loss with no effective treatment. So our goal is to develop a bioinformatic pipeline to explore the role of alternative splicing in PAD based on high-depth RNA-seq, as described in Figure 2. To this end, we implemented a multi-pronged approach to uncover splicing variants. We used isoform-based methods that seek to reconstruct full-length transcripts and estimate their abundance, exon-based methods that are biologically less relevant than transcript-based approaches but computationally more reliable, and event-based methods that seek to quantify splicing events by calculating the percentage spliced in, or PSI values. Because differential isoform usage based on short-read and RNA-seq suffers from false positive bias, we look for consensus across all approaches. So based on our preliminary results, we find 275 exons that correspond to 201 transcripts and 150 unique genes to be differentially spliced between PAD and non-PAD controls. And of those, 13 genes are also found to be differentially expressed overall and shown in the table in the center panel. Top 10 pathways from the gene set enrichment analysis based on the 150 differentially spliced genes are also shown below. And as a case example, the top of the panel on the right shows one protein coding gene for which that's differentially expressed at the overall gene level and is higher in PAD than controls, but where one transcript appears to be differentially utilized and higher in controls than in PAD. And this is confirmed using two different transcript-based tools, RSEM and Callisto. The panel at the center panel on the right shows results from DEXIC, which is an exon-based method that finds the same transcript also differentially utilized at the exon level. And in all these analyses, the transcript's differential utilization appears consistently higher in controls relative to PAD. Using RMATS, which is an event-based method, we also found that this transcript has a significant exon skipping event. So although conceptually useful, exons are ambiguously defined. As an example, this gene appears to have 12 exons according to Entree, but 22 according to Ensemble. So as an independent data-driven approach, we also propose to consider the average number of read counts per locus detected in cases versus controls, which may define de novo differentially utilized transcript regions. That's work still in progress and is illustrated for this particular gene on the bottom figure on the right panel. With that, thank you so much for listening, and I'll be happy to answer in the chat any questions you may have. Thank you.